Hi, I hope everyone's doing well. I'm Ellen, and this is Anna and Aoife, and we're from Parkside 6th, which is an international sixth form from Cambridge. And our Authentic Biology Project is looking at the isolation and characterisation of bacteriophages.
So today we're going to address two questions. First of all, what bacteriophages are, and secondly, why they're important. So bacteriophage is a virus which parasitizes a bacterium by infecting it and reproducing inside it. They're used much in genetic research, and here you can see the structure of a bacteriophage. So its genome is embedded into a complicated protein skeleton, and the genome has all the instructions on how a bacterium can make a new bacteriophage. This is an electron microscopic image of the bacterium being attacked by many bacteriophages. The white dots are the bacteriophages. So now some facts about these viruses. So they were discovered in 1915 by Frederick Twors, a British pathologist, and in 1917 by Félix Terrel, a French-Canadian microbiologist. They play a key role in driving the evolution of bacteria, as bacteria have evolved many ways of avoiding lethality, and this is why we can call this process a molecular arms race. So bacteriophages are extremely abundant, about 10 times more common than their bacterial hosts. But bacteria are still around because they have evolved clever strategies to avoid the lethal impact of the viral parasites. One of these strategies is the use of restriction enzymes. These are produced by certain bacteria, and they have the ability to cut the sequence of bases at specific sites, and hereby they protect the bacterium. So bacteriophages can replicate two ways. One of them is a lytic cycle, and the other one is the lysogenic one. So we focus on the lytic cycle because the lysogenic cycle can take a very long time to complete. And both start off when the bacteriophage attaches to bacterium and infects it by injecting its genetic material inside it. The next step is the production of phage nucleic acid and protein coats. So when the new bacteriophages, the mature ones, have assembled, they cause the cell walls to burst, and this is called lysis. And in the end, the bacterium dies.
So our process throughout 2 years has 5 key steps. And in the first year, we isolate and purify new strains of phages by collecting samples of the phages ourselves. And in the second year, we carry out the final 3 steps, which is identifying the structure of the phages using electron microscopy, extracting the DNA from the phages, and identifying the genes. So the first step is isolation. Now, bacteriophages are very easy to collect. Wherever you find bacteria, you'll find bacteriophages as well. And we collected our samples of bacteriophages from the River Cam, which is a local river in Cambridge. And each of these samples contains billions of different strains of bacteriophages. And in order to get only bacteriophages in the sample, we had to filter these. And we infected a bacterial host. We used E. coli. And then we mixed this mixture with top agar and poured it onto a Petri dish. And we allowed the bacteria to multiply and the bacteriophages to replicate on the Petri dish. And the bacteriophages kill bacteria and neighbours on the plate. As you can see on the picture, the bacteria leaves a cloudy lawn on the Petri dish, and the bacteriophages result in a visible clear circle, which we call a plaque. And the next step is purification. In the first infection stage, there are many different types of bacteriophages. So we want to purify them so we only get one kind of bacteriophage. So what we do is we pick a single plaque from the plate and repeat the infection and plating process 3 times in order to purify the stock.
Okay. So stage 3 is where the structure of these bacteriophages is analysed, and we use electron microscopy to do this. And what we've discovered is the extent to which the structures of different bacteriophage can vary. So these images show how the tail lengths of bacteriophage can be vastly different, and this range of structures could be reflective of the diverse range of bacteriophage which have the ability to target and destroy particular strains of bacteria. So the long-term goal of this structural analysis is to understand the implications of structure on bacteriophage behaviour. So the 4th step was to isolate the DNA of these bacteriophage, and this was all done using standard procedures. The data collected was then sent off to be sequenced. By finding the genome of the collected bacteriophage, the genetic makeup and the structure of the bacteriophage could be related. So stage 5 is bioinformatics, and this is, so when the sequenced DNA is returned, we input the sequence into a program called Artemis. The data is then blasted, BLAST being Basic Logical Alliance Sequencing Technique. I'm not too familiar with it myself. And similar genes are returned, and we can annotate our genes using this and using the Artemis program. So the following, we blast, identify similar genes and annotate. So the following are the results that we've received from previous years, over the last year, and they show varying presences of tRNA. So B isn't complete because we haven't annotated it yet, but the rest, basically, if you can't understand these numbers, the intention is to show you that there is a variety of different ways in which these bacteriophage can come around. So different structures of bacteriophage are all able to target particular bacteria. We've just received our sequenced DNA, and we will be beginning the bioinformatics process this year. So all of these steps contribute to our understanding of the genes involved in shaping the bacteriophage structure. The project allows insight into the genetics of this set of viruses. Long term, one of our aims is to develop an understanding of how and potentially why these viruses affect specific bacteria. But of course, like all the other projects here today, the overreaching target of this project is in its social implications. Our hope is that one day this information we collected can be utilised in the production of alternatives to antibiotics, with antibiotic resistance becoming increasingly prevalent. None of this progress we have achieved or the future advancements of this project would be possible without the constant support we have been given. And as a result, we would like to thank the Wellcome Trust for hosting us today and for their continued generosity and support. We would also like to thank the University of Cambridge, particularly the Biochemistry Department, for allowing us to use their labs and equipment. We thank Parkside 6th, all the students and teachers involved, and we thank you all for listening.
Thank you. Got to be careful dipping in the River Cam, you know. Some stuff in there you don't want to touch. Right, do we have any questions? There we go, perfect.
Was there a reason why you chose E. coli as your host?
Yeah. Well, it was the least harmful one to use in a school, really. We chose it out of mostly convenience. But as to the full reason why we chose it, our teacher knows that better than me. So I can only say it was out of convenience. Yeah.
Do you think it will be interesting to look at other hosts? Presumably some of the phages you've isolated might prefer other
I think
hosts?
that's definitely an extension that could be undertaken looking at the way different bacteria interact. But currently we're just looking at how you can get multiple different sort of structures for bacteriophage, and they can still affect one particular strain of bacteria. So,
Thank you.
Cool. Thanks. Right, any more? Just do you want to ask yours, Becky? I don't think you need a microphone, but, you know, just for the recording.
I just want to say, it's fantastic. Are you going to be part of the whole... Is this a major part of the sort of the quest to try and find new antibiotics? Are you, collaborating with any of those other players in that field? Or is that part of how you link with Cambridge?
We've been pretty fortunate to have a lot of interactions with the university, so working in their labs to isolate the DNA and then running it through an electron microscope ourselves. But we're still working on the networking with other universities and more people to do research, but currently it's sort of an independent project that's run through the University of Cambridge, and they can use the data that we find any way they please.
And I have a question to chuck in for the other two. You mentioned antibiotic resistance a little bit at the end there. Can you kind of expand on that a little bit? Are you concerned about that in the future, and what do you think this research could contribute?
So yeah, I'm sure that you've all heard of antibiotic resistance. People not taking the full course of their antibiotics, so the most resistant bacteria stay towards the end. And these bacteriophages could potentially be an alternative to antibiotics, which is really useful, and I think we all feel quite privileged to be able to be a part of this. So yeah.
Great. Thank you so much. Give them a huge round of applause. Thank you.